Every year, 50 million people get diagnosed with a neurodegenerative disease around the world. Because of that, people tend to lose their ability to speak, walk, or remember things as the cells in their brain get progressively damaged. Currently, there are no cures available, only medications which can help to manage the symptoms. But this could become the thing of the past if we start to use neurospheres. Hello everyone, I'm Aliya and I'm interested in regenerative medicine, particularly how we can use stem cell therapy to regenerate the damaged areas of our nervous system. So, neurospheres are basically spheres that are made up of different cells, and this is what they look like, except they don't have any spikes, are a million times smaller, and aren't blue. This is just the closest thing that I could find at home. Here's a real picture of them. What makes them so unique is that they have this type of cells inside called neural stem cells. These are special cells which can differentiate or become specialized to turn into cells of our nervous system. Therefore, because they have this unique ability to turn into the cells of our brain or spinal cord, for example, they can be used in patients with Alzheimer's, Parkinson's disease, or spinal cord injury, which can then help them to replace the damaged cells in their nervous system. As a result, millions of people would be able to walk or speak again, but we can't just get them from pharmacies. And in order to do that, we need to proliferate either neural stem or progenitor cells in labs in serum-free conditions using the fibroblast growth factor and epidermal growth factor. Let me explain. It has been observed that neurospheres develop from one cell called the founder cell and consist of its progeny. The founder cell acts as the starting point of neurospheres development, and then as it proliferates or divides to increase in number, the resulting cells are its descendants or progeny, which will eventually make up the neurosphere. One of the biggest conditions that we have to follow when developing these neurospheres is that there shouldn't be any serum used. Serum is basically a substance found in blood that can help to promote growth. But in case of neurospheres, the epidermal growth factor and fibroblast growth factor is used to prevent contamination and promote growth. So, once you have all the required conditions to develop the neurosphere, then after about 7 days, you may have neurospheres with a diameter of about 100 to 200 micrometers. But this can also depend on the conditions of the experiment, and therefore it can take up to 2 months to see any substantial growth. But other than the experimental conditions, the type of founder cell can also affect the size of neurospheres. Neurosphere size is an important factor to consider if we want to use them in medicine or research, because if the neurosphere is too big, then it may be hard for the innermost cells to get enough of oxygen and nutrients, and therefore it can affect their functionality and health. Smaller neurospheres tend to promote proliferation, which is increase in cell number, while bigger neurospheres tend to promote differentiation, which can help us to get the right cell type that we need. Also, if we want to use neurospheres in medicine, then it is important to consider which size would be the best option for us to transplant into patients' bodies. And as far as we know for now, smaller neurospheres are a better option because they're easier to deliver to the targeted areas and they're easier to integrate to the existing tissue. So that is why I decided to test how the type of founder cell affects the neurosphere size. To do that, I used the neurosphere simulator developed by Northeastern Neurobiology, and here are my results. So what I did was that I counted the number of live cells in each case and repeated the same experiment 10 times to get reliable results. Now, if we do the calculations based on the results from my table, the average number of live cells when stem cells act as the founder cell inside the neurosphere are 54 live cells, while the number of live cells inside the neurosphere when progenitor cells are used as the founder cells, it is 38 cells. And therefore, we can say that when progenitor cells are used as the founder cells, the neurospheres are indeed smaller. For those of you who are interested, I'll show the simulator at the end of the video. This can be explained by the fact that progenitor cells have a more limited ability to self-renew, and so form new cells, decreasing the size of neurospheres. Also, it has been observed that when progenitor cells act as the founder cell, there is a high proportion of progenitor cells inside them, which means that you'll have more cells with a limited ability to self-renew, decreasing the size of neurospheres even further. So you may ask yourself, which option is better, larger or smaller neurospheres? Well, it depends on what you want to use them for. Larger neurospheres are a better option if you want to study intrinsic specifications. These are characteristics of cells that help to determine their development and function. Also, they're a better option if you want to study quantitative statements or the number of precursor cells inside neurospheres. These are special cells that are able to differentiate only into one single cell type. Smaller neurospheres, however, are a better option if you want to study differentiation. Smaller neurospheres can be easily dissociated, which means that their 3D structure can be easily broken down into individual cells. This can help to make the studies of differentiation easier, and they could potentially act as the source of cells for patients in regenerative medicine. Also, as we've seen, progenitor cells, when they act as founder cells, tend to form smaller neurospheres. This means that you will have a higher proportion of functional progenitor cells inside neurospheres, and this can be beneficial because progenitor cells are better at integrating with existing tissues and cells, especially in damaged areas. And so it is up to which option is better for you. To conclude this video, I'd like to point out that neurospheres could become the key to treating neurodegenerative diseases and also injuries. But before we actually do that, there is still a lot of research we have to do to understand how to implement them in real patients and also understand potential side effects that may happen long term. Thank you for watching, and until next time. Hey guys, so thank you for watching the video till the end, and as promised, here is the neurosphere simulator that I was talking about throughout the video. So for this experiment, as I said, we will be focusing on the founder cell type and how it affects the development of the neurosphere. And so here, to do that, we will repeat it 10 times to get reliable result, as I said. And here, as you can see, you have different parameters that you can choose and change to see how they will affect the development of the neurosphere. But for this experiment, we will be focusing only on the founder cell type. So we will repeat the experiment 10 times, as I said, with each cell type. So like here, just to give you an example, I'll leave it as an active stem cell, and I'll select the large lattice size to give it more space for growth, and the rest of it will leave as it is, so the default settings. And here I'm gonna run the model, and depending on how fast your internet is, the diagram and the graph should show up, or I will show you. It shows how the neurosphere develops. So as you can see, here it shows like the development of it, as I said, over time. And here we have like a diagram that shows the type of cells that the neurosphere has inside it. And here we have the cell population size. So the cell population of each cell type, and here like we can see the key to the different colors and what exact cell type do they represent. And so to count, for the experiment, what we do is that we count the number of live cells. So every single cell that we have here, except the dead cells, which are in black, but here we don't have that. So we just count it, and then you repeat the experiment 10 times, and you take the average number of live cells as I did. So for the large neurosphere, for the active stem cell, it was 47 live cells, 54 live cells, and for the progenitor cell, it was 38. And yeah, thank you once again. Thank you for watching this video, and until next time, bye.